isolate a lot of proteins, and here is a kind of guide to some of the main techniques that you might see when you want to express or make and purify proteins. And so I want to take you on like a walkthrough of a typical workflow, everything from cloning to going through all those chromatography steps, concentrating it down, flash freezing it, and sticking it in the -80 for storage. And so I have a whole page of posts on topics of these topics, and so I will kind of just walk you through that page, as well as then you can go and check out these posts for more information. So I hope that this is helpful, and let's get started. So just an overview of where we're going. We're going to be talking about how we can get cells to make specific proteins through recombinant protein expression and purification, as well as some troubleshooting things. When we're dealing with protein purification, we're often dealing with protein chromatography, where basically we separate proteins based on their different properties by sending them traveling through these columns filled with these little beads called resin. And because proteins have different properties and they're going to interact with these resin in different ways, and therefore this is going to alter how they flow through that column. And this is going to allow you to separate them based on things like whether or not they have a specific tag, or what charge they have, or what size they have. And so we'll go through some of these common chromatography techniques that we use. These techniques, most of them can be used for both if you're purifying something native, so something the cells make themselves, or whether you're dealing with something recombinant. So basically this is something that the cells wouldn't normally make, but you give them the instructions to make it, and so they will. And so we'll talk a lot about about molecular cloning, how we can actually design the genetic instructions that we can put into these cells to get them to make it. We'll also talk about some of the cells that we can stick it in. So often this is in bacteria, but this doesn't always work. So there are some tricks that you can try in order to try to get bacteria to produce it better. But you might have to move on to other types of cells, such as insect cells. There's also like cell-free expression systems, which you can do some cool things like incorporate non-canonical amino acids, so kind of non-normal protein letters that can have specific cool things on them, like labels and things like this. Also talk about no matter where your protein is coming from, whether it's recombinant or native, you're going to have to isolate it out of those cells. So we'll talk about how we can first isolate those cells in the absence of the media. We call this like harvesting. How we can break open those cells. And then the purification stuff we talked about, as well as some things like dialysis, where you're trying to get rid of extra salt, such as when you use one of those techniques where you have to push the things off the columns with a lot of salt to compete them off. There's also like desalting and buffer exchange gel filtration columns we'll talk about. Before you store your protein, you're going to want to concentrate it, and you have to concentrate it at certain steps in the purification process as well. So I'll talk briefly about how we can concentrate proteins with these centrifugal filters, how you can measure the concentration of those proteins, how you can check the purity of those proteins and see what's in your mixture, and finally how you can flash freeze that protein and keep it nice and safe in storage. As to what you can do with that protein, so yesterday I made a big or a couple days ago when I'm posting this, I made a post all about my lab techniques page, where I have a lot more information about various techniques. And if you want that you can do to measure how this protein interacts with other proteins or with nucleic acids, things like this. Sometimes what we want to do is we want to look at the protein at the atomic level, so we can use structural biology techniques like exocrystallography and cryo-EM, and so I have a lot more on those on my structural biology page. But today I want to focus on the protein expression and purification. So I'm going to put timestamps on the video so you can jump to where you want to go. But I'm going to start by talking about protein chromatography. This is going to be applicable no matter what type of where your protein is coming from, no matter what type of cells it's coming from, whether or not it's recombinant. So it's something that is getting made from the genetic instructions you stuck in there, or whether it's something that these cells are just naturally making for themselves and you're just trying to purify out of them. So after I talk about protein chromatography, then I'll go and talk more about the recombinant expression aspects. But let's start with this protein chromatography. So there's actually multiple forms of protein chromatography, and typically what we're doing is we're doing multiple steps of these chromatography columns. So as I mentioned, these columns are filled with these little beads called resin, and there are different types of resin. And these different types of resin are going to interact differently with different proteins based on the different properties of those proteins. So lots of different things. Basically, some of these resins, they might have charge. And so if you say have negatively charged resin, it's going to bind to positively charged parts of proteins. And if you have positively charged resin, it's going to bind to negatively charged parts of proteins. And so we're going to see how we can take advantage of charge using ion exchange chromatography. Another thing that proteins have is different sizes. And so we can use size exclusion chromatography. Here the resin, it has pores of different sizes that are going to separate the proteins based on their sizes, as we'll see. So these can take advantage of natural properties about proteins. But often what we can do is if we're doing recombinant protein expression, since we can manipulate the genetic instructions, we can get it to add the cells to add on an extra tag on the end. So add on a few extra amino acids that are going to bind specifically to this affinity resin. And that's going to allow you to kind of do a capture step where you can purify out your protein from most of the other stuff, because that other stuff isn't going to have that special feature that this protein only has because you added it on. So all of the proteins are going to have some sort of charge, and all of them are going to have some sort of size, but not all of them are going to have that special feature. So we often start with one of these affinity chromatography steps, something like a his tag or a strep tag, to capture our protein, then follow that up with taking advantage of, say, the charge and the size. And so it's not that you're just using a single chromatography strategy typically, but if you want something really, really pure, you're going to go through multiple steps. Depending on how pure you want your thing to be, you may or may not need to use multiple steps. But anyway, let's start with this affinity chromatography. So if you're using affinity chromatography, this is typically going to be your first step because it's going to get rid of the most stuff, because it's taking advantage of that thing that's super specific about your protein. This doesn't mean it's going to get rid of everything else. There's going to be some non-specific binders, but it's going to get rid of most of the stuff. And so you want to take advantage of this first. And so there are different types of affinity chromatography based on different tags we can put on our protein. And there are also some forms of affinity chromatography that take advantage of natural features of a protein, but that are very specific features of that protein. So it might be, say, like, if your protein binds DNA, you might have like double-stranded DNA or something. You could have a column with the resin was attached to double-stranded DNA or something like this. But usually when we're talking about affinity chromatography, it's on some sort of engineered tag that we've put onto the end of our protein. And by tag, remember, it's just like a few amino acids that we put onto the end. Or it could be even like a bigger protein we put onto the end, such as like a fusion partner we'll talk about, like GST or MBP. But often it's just something short that's not going to interfere too much. So you might even be able to leave it on your protein. But you can also often have like a protease cleavage site, so you can cut this tag off once you've used it if you don't want it hanging around. One of the most common tags you'll see used is a his tag. So typically this is like 6 to 8 histidines in a row. What these are going to do is these histidines can act as like a chelator or metal biter. And they can bite down around metals like nickel. And so in this IMAX, so this immobilized metal affinity chromatography, we have a metal, so typically nickel, sometimes cobalt, immobilized or like attached to the resin in these columns. And now your protein, if it has that his tag, that histidine tag is going to basically bind really tightly to that metal. And so it's going to bind to that metal, and that metal is bound to the resin. And in this way, your protein is kind of indirectly bound to the resin. You can get your protein to bind, wash everything else off while your protein is still bound, and then elute your protein. So basically get your protein to come off by competing it off with imidazole, which basically is a competitor that looks like histidine. It's kind of just like this part of histidine, but it's free floating. And so we can flood the column with a bunch of imidazole to get our protein to come off. And so this is a common strategy that we use, this IMAC, with this his tag purification. So this is often great for bacterial protein expression, but they do it might be not as great for non-bacterial. Conversely, a strep tag can be really great for non-bacterial, but less great for bacterial because bacteria use more like biotin-like things. So what is this strep tag? This strep tag is basically another different sequence of amino acids. This one's a little more balanced. And what it's going to do is it's going to be a mimic of biotin. And so it's going to bind to a mimic of streptavidin. And so you basically have this streptactin resin, which is going to bind to the specific strep tag. And then similarly to above, we're going to wash everything else off. But in this case, we're going to be eluting or competing off our protein with this thiobiotin. So very similar strategy with just a different tag. There are also other tags we'll talk about that can serve as fusion tags. And so they can act kind of like solubility helpers, such as GMP and MBP, but they can also then act as affinity tags. So with glutathione, with a GST tag, you can bind to glutathione and things like this. So different affinity strategies. For this, I'm often doing this in batch mode. So basically just gravity flow in a column like this. You have kind of bind your protein with the resin, either in like a batch. So you do the binding in a tube and then you stick it in, or some sort of bottle and stick it in, or you can just flow it through the column. You can also do this on a protein purification machine, and some of the next steps are a lot easier to do on a protein purification machine. So when we talk about these protein purification machines, often it's an AKTA. That's a brand name, but it's a form of this fast protein liquid chromatography, or FPLC. Basically what it does is it's going to send buffers or these pH stabilized salt waters that we want to purify our proteins with. It's going to flow these through the column using pumps, so you can really control the flow rate, as well as if it has like a sample system. These system pumps can kind of mix and match different buffers in order to do things like make gradients, where you increase the salt concentration, or you increase the pH, you increase the concentration of the competitor. You can do this various things. Then your sample is going to come out through the column, go through a UV detector, and then into a fraction collector. So it's going to collect little portions. And these portions, you can look and see where they correspond to the chromatograph. So to when the UV what the UV is detecting, because the UV detection can show you where proteins are coming out. And then you can compare that to the different fractions to take the fractions that you want to use. So this is really great when you want to do ion exchange chromatography, that you can have a gradient be made. You can have a gradient, like a salt gradient, that's going to allow you to compete off proteins that are bound based on their charge. So this is a strategy with ion exchange chromatography, is that different proteins have different charges. And instead of like overall charge, that's a good place to start, is looking at the overall charge. But really what's going to matter the most is have like local regions of charge. So you kind of have charge patches on the protein. And these charge regions of the protein are going to bind to oppositely charged resin. And so in cation exchange, what you have is you have a negatively charged resin, and then a positive or cationic protein. Conversely, with anion exchange, you have a positively charged resin and a negative or anionic protein. What you're going to do now is you're going to bind that protein to the resin. And in order to get your protein to come off, we're going to use a salt gradient. So a salt is just a neutral combo of a cation, so of a positively charged thing, and an anion, a negatively charged thing. And so if you stick a salt in water and it's soluble, it's also going to dissociate. So if you stuck table salt, so NaCl, into water, you get sodium and chloride ions. Now the sodium ions are positively charged, so they're going to bind to that negatively charged resin. And the chloride ions are negatively charged, and so they're going to bind to the positively charged resin. And so in either case, when you put that salt in there, you're going to get these salts kind of competing for positions on the resin. The more salts that you have, the more it's going to outcompete your protein. So even if your protein is stuck on there, the more salt you add, your protein is gradually going to get pushed off. The more oppositely charged your protein was, the tighter it will bind, and therefore the more salt it will take to push it off. And so we can increase the salt concentration gradually in order to get proteins to come off based on how strongly they were stuck. So this is a strategy we can use to separate proteins based on their charge. We bind them to oppositely charged resin and then gradually increase the salt concentration. The ones that are most weakly bound are going to come off first. The ones that are more strongly bound are going to come off last. And then we can, hopefully ours is going to be one of the most oppositely charged ones. But even if it's not, what we can do is we can then test those different fractions out and see where they, see which ones had our protein. So at this point, what we've done, if we've done the affinity capture step, we captured most of it. We might have cut the tag off afterwards. We didn't need it anymore. If it's a big tag, we often really do cut it off. Something like GST or MBP that might actually make your protein dimerase. So basically form these pairs, team up in pairs, which you might not want. Or this tag can get in the way. If you have a shorter tag, it might not be a big deal. But either way, you often have like a thrombin cleavage site or a TEV cleavage site, something like this. You can cut the tag off. Once you've cut that tag off, now you have to take advantage of natural things about the protein, such as their charge. So we've done ion exchange chromatography. Another natural thing about a protein we can take advantage of is their size. And so this is what strategy we use in size exclusion chromatography. This is sometimes called gel filtration, and we can do it. Basically, what it's going to do is it's going to separate proteins by size using these kind of boring columns, boring resin. So I say boring because they're like they don't have charge, they don't have special groups on them. But also, I guess you can kind of think of boring as in there's tunnels bored into them. These pores are basically formed, these resin they basically have these pores. So it's not like they're really like bored into them. They just happen to be, they're kind of like these gels, kind of like your agarose gel, just in bead form. And so they have these little kind of like secret tunnels flowing through them. And these tunnels, basically you choose resin with different size tunnels. And so depending on the size of the things you want to separate. How it works is that the bigger things aren't going to be able to go through the small tunnels, whereas the small things are going to have to go through all the tunnels. And if you have to go through all the tunnels, you're going to be facing a longer travel route. And if you have to go a longer route, it's going to take you longer, even if you're flowing at the same like if you're flowing at the same like speed, you're going to be going further than the bigger things, and so you're going to take longer to get through the column. And so in this way, the bigger things are going to come out first, the smaller things are going to come out later, and you can choose what size resin you want based on the size of the things that you're trying to separate. So we have a bunch of different columns that we can use based on the sizes of the things we want. And these are going to be things like your superdose, your superose, your superdex, your dextran, various things like this, and they come in a variety of sizes and shapes and things like this, and much more on it in my size exclusion chromatography post. When you're working with one of these machines, you're going to be having to read that UV trace, that chromatogram, and so I have a post on how you can do that. So basically, this is just taking advantage of the fact that proteins absorb UV light of a 280 nanomolar wavelength, nanometer. I always say nanomolar, but nanometer wavelength. So you can monitor protein coming off, and then cross-reference to the fractions that are being collected in order to see where your protein probably is. So those are more like serious versions. I like to think about protein chromatography as kind of sending proteins on field trips through museums. And so if you want to see the museum analogy of protein chromatography, I have a post on that as well. So now let's back up and talk about how we actually get that protein to be made in order to purify it. So often these days we're doing recombinant protein expression. So remember, recombinant, we're recombining bits of DNA. We're taking the genetic instructions for making our protein, so we're taking the cDNA, and we're sticking it into a plasmid, so a circular piece of DNA, that we can then use as a vector or vehicle to stick into cells and get those cells to make protein for us. So here it says like your gene, but really what we're talking about is the cDNA, the complementary DNA. This is going to be a DNA version of the edited version of the messenger RNA of the gene. And so basically in transcription, a messenger RNA copy of a gene gets made, but really it's a pre-messenger RNA copy, and then the regulatory regions, the introns in this gene get cut out and the exons spliced together, so you get this you get this edited version that doesn't have the regulatory information that would just confuse the ribosome. So it would confuse the protein making machinery. And so what we stick in is cDNA, which is a DNA version of this edited mature messenger RNA. So just a technical note, and I post on that as well. But we stick that into this vector. And before we stick this vector into cells to make the protein for us, we actually stick it into like cloning cells that we're using to make lots of copies of it, and then make sure that the sequence of it is all okay. And so I have a lot more posts on molecular cloning, how we can use strategies like SLIC and restriction enzyme based cloning in order to get this plasmid made, and then how we can verify that we made it correctly, and then ultimately how we can stick it into expression cells to get the cells to make the protein for us. As I mentioned before, we're often sticking some sort of affinity tag onto the end. We can also do things like stick on a fusion tag, which might also kind of double as an affinity tag. And so some common fusion partners are SUMO and GST and MBP, NUS-A and TRX. What these have in common is that they're these little proteins that the cells really like to make, and they're really soluble. And so sometimes the cells don't like to make your protein, or your protein might have trouble folding and staying soluble, things like this. And if you stick a fusion protein onto the end, something that these cells really like making, then you can kind of trick them into making your protein, and it'll help keep your protein soluble as well. And then often, because these are going to be a bigger protein on the end, you're going to have a protease recognition site, so you can cut that fusion partner off after you've used it, and hopefully your protein still stays soluble. So much more on this in this post on fusion partners, but I just wanted to mention that here. So now you've cloned your thing, you have whatever tag you want added on it, you have a fusion partner potentially. Now it's time to stick it into cells, and you have to choose what type of cells you want to stick it into. Well, actually, you had to choose that when you did all that cloning steps. So you stick it in a plasmid that's going to be compatible with the cells you want to put it into. When it's possible, we often use bacterial cells. This is going to be like your cheapest and easiest method. Bacteria, we often use this overexpression system with this T7 system. I have a whole post on it, but basically what happens is you can get the proteins to get the cells to make your protein on demand by adding a chemical called IPTG. It's really cool how it works. Basically, the IPTG is going to cause the proteins to start making T7 polymerase, and that T7 polymerase is then going to make lots of messenger RNA copies of your protein, which you've put under the control of a T7 promoter, and then those mRNA copies are going to outcompete the bacterial copies and on the bacterial messenger RNAs, and so you're going to get lots and lots of your protein made. So we say we're like overexpressing it. We can actually get these cells to devote like most of their energy to making your protein, and end up with having like half the protein in the cell be your protein of interest, which is great as long as your protein isn't toxic to the cells. But another great thing about this is that you get to control when the cells make the protein, and so you can make it so that they don't grow that long and things like that. But basically this T7 expression system is going to be really great. But sometimes the bacteria aren't very great at making your protein, especially if you're trying to do too strong of an induction or something like this. If the bacteria are kind of struggling to fold your protein, you might overwhelm them. Even if you're not doing too intensive induction, you still might have problems, because as I'm not sure if I mentioned this before, but basically bacteria, they don't have all the same machinery as our cells do. And so they don't have the same like glycosylators and phosphorylators and things like this. They're like kinases and things that are going to modify your protein in specific ways. The bacteria might modify the protein in different ways. And they also have different chaperones and things like this. So basically chaperones are going to help the protein get folded as it's coming out of the ribosome. And if you have suboptimal a protein that is like kind of complicated and needs help folding, it might not work well in the bacteria. But sometimes you can get things to work in bacteria even if it's not ideal for the bacteria. One of the ways that you can do this is with using codon optimization. Basically, the genetic code is degenerate, meaning that there's multiple ways to spell the same amino acid. So you can have different messenger RNAs that are going to make the same protein. And some of those messenger RNAs are going to be more ideal or more optimal for a specific cell type based on what that cell type usually uses, and so what they have the most of in terms of their tRNAs. which correspond to the codons and bring the amino acid that corresponds to the codon in the sequence. And so much more on that in other posts, so I'm not going to get into it too much here. But just note that you can use codon optimization to try to change the sequence without changing the messenger RNA sequence. So change the DNA sequence, which will change the messenger RNA sequence without changing the actual protein sequence. And so there are tools that you can use for this codon optimization. And then you can order like a codon optimized version of the sequence. Another strategy is to basically just add extra genes for making those rare tRNAs. This is the strategy with things like Rosetta strains. There are also a bunch of different strains of bacterial which might be better or worse for making different proteins. And so a common strain that we use for expression is BL21. The BL21DE3 version has the T7 RNA polymerase under that lac promoter, so you can use that inducible system. And there are other versions of BL21 as well. And I will leave you to check out that post on your own if you want. Sometimes bacteria just aren't cut out for the job. So sometimes we might want to turn to cells that are eukaryotic, so cells that are more like our cells. They have membrane-bound compartments. They have their DNA kind of housed up in the membrane-bound nucleus, things like this. And so if we want to turn to eukaryotic cells, we often, different common ways are like yeast cells, insect cells, and mammalian cells. And so when we're I've never actually used yeast cells. I've heard that they're really hard to break open. I've used insect cells. And so with insect cells, you can use this baculovirus expression system, this back-made expression vector system, or BEVS. This baculovirus expression vector system, or BEVS. And basically you make this like virus that infects these insect cells, and then these insect cells start making the protein. And so much more on that in this post here. With mammalian cells, we're often dealing with like a CHO line, so Chinese hamster ovaries, or an HEK line like freestyle or XB cells. With any of those, you're typically working in suspension, so you have media growing in like flasks. You can also work with like cell-free protein expression, like small scale or larger scale at like companies and things. This can be based on a lysate, so based on like cells that are broken open, and then you just use their guts and add stuff to it to make it more efficient. So this can be like bacterial lysate, wheat germ lysate, rabbit reticulocytes, or other cell lysates. There are also systems that are reconstituted from pure components, such as the pure system, where you basically, this is an E. coli system where you have all these different components that are needed for translation for protein making are purified and mixed together. And then this is really great if you want to test out specific things, make modifications, but this is typically done in like a smaller scale reaction. There's a lot of work being done, though, to try to make larger scale versions of things like this so we can do more cell-free protein expression at larger scales. One of the really cool things about cell-free protein expression is that you can, and especially with like the pure system, is you can use unnatural or non-canonical amino acids, so protein letters that we don't normally have. You can have modified versions that might have things like labels. This is going to allow you to do a lot of cool chemistry, things like bioorthogonal chemistry and click chemistry and stuff like that, where basically you're using chemical reactions with different chemical groups and things like this that our cells don't have, so you get specificity for the modified versions that you've put into these cells, but you're still able to interrogate or kind of use have the cells do what they would normally do, but with modified things, and that's going to allow you then to get the cells to do non-normal things, or to at least track what's going on, various cool things. And so check out the post on that for more. No matter where you're getting your protein made, well, as long as it's not cell-free, you're going to have to break open the cells to get the protein out. But when you break open those cells, you want to make sure that they're in a nice, safe environment. You don't want to just break them out into the media that they're growing in, so into like the food that they're growing in and secreting all their waste into and this sort of stuff. You don't want to break them out into that. Instead, you want to break them out into a nice, clean liquid with protease inhibitors so that they're going to stop those proteases, so protein chewers, from chewing up your protein. And so typically what we do is we actually harvest the cells. We basically collect them. We spin them down so that the cells get separated from the media. We remove the media. Then we might wash the cells. So basically stick in nice clean liquid, like some sort of buffer with protease inhibitors, and then spin them again to wash them. Or we might just pellet them, or we might just like freeze the pellet as is. Or we might resuspend them in the buffer and then freeze the resuspended buffer. Basically, we have a lot of options. But no matter what you do, you want to basically put it into a smaller volume without the media, and potentially with a buffer that's going to be safe for them when they break open. Or you can just add the buffer later. In terms of how you actually go about breaking them open, this process is called lysis, and this is also cellular disruption. There are different methods. Some of them are like physical methods, so like freeze thaw, sonicators, homogenizers, French presses, things like this, microfluidizers, bead mills. Then there are enzymatic methods, things like using lysozyme to help you break open the bacterial cell wall, and chemical methods such as using detergents. And one of the common methods we use is ultrasonication, where basically we use these ultrasonic waves to kind of send these waves traveling through this liquid. And these waves are going to then, like in their wakes, they're going to form these bubbles, and these bubbles are going to burst. It's called like cavitation and gaseous cavitation, and it's going to release shock waves that are going to help break open the cells. And once you've broken them open, then we go back and we do all that protein chromatography. At various steps in the protein chromatography, we're using really high concentrations of salt. So this might be like imidazole that you're using to compete off your protein from your his-tagged protein from the IMAC. It might be high salt that you're using in ion exchange. But sometimes you want to remove this salt. So for example, if you're using a high amount of imidazole in order to elute off your his-tagged protein, and then you want to go and you want to do ion exchange, that high salt is going to interfere with your protein binding to the column, and so you want to get rid of it first. A common way we can get rid of excess salt is with dialysis. Dialysis is going to take advantage of semi-permeable membranes. So basically, these are going to be membranes that are going to allow some things through, but not others. So you choose a membrane that's going to keep your protein in it. So your protein can't go through the membrane, but other little things such as the salts can. And so you can stick your protein in this membrane pouch, and then the salts are going to flow out thanks to diffusion. And you have you make it so that you have a low concentration of salt outside of the membrane, and so the salt is going to diffuse out, and then you replace that water, and you're going to gradually remove all of the salt, or at least most of it. And so this can be a kind of slow, tedious process. Another option is going to be like a desalting column, which is going to be like a size exclusion column, except that the pores are going to be really tiny. And so most of the stuff is not going to go through, but those little salts are. And so your protein is just going to go straight through around those beads. And so you can separate them from the salts that gets trapped. So you might be using something like a PD10 column for that. Other times in the protein purification, if you want to, before you say go do that size exclusion chromatography, you're going to want to concentrate it, because in that size exclusion chromatography, things are not going to get stuck on the column. They're just going to kind of flow through the column. So you don't want things to have a head start like you would have if you had a large volume. Some of the proteins would be entering way before the other proteins, and so you would think that they were bigger, but really they had just gotten there first. And so you want to concentrate it to a small volume before you inject it. Even if you're not doing that, at the end of your protein purification, you're going to want to concentrate it, because the proteins are going to be more stable at a higher concentration. And as I talk about in my post on storing solutions, if you have a higher concentration solution that you're storing, you're going to have less proportional loss based on sticking to the sides of the tube. So a common way that we do this concentration is with centrifugal ultrafiltration. So basically we have these spin concentrators where they have a membrane. They have this kind of little bucket on the top of this membrane. So kind of like what we talked about in dialysis, the protein can't go through, but the little stuff can. And so basically your protein's going to get stuck up here. When you centrifuge this, it's going to kind of help pull the liquid through as well as the little things, but your protein's going to stay stuck up top. And so in this way, you're able to concentrate it. You want to be able to measure how concentrated it is, and there are a bunch of different methods for measuring protein concentration, and I have posts on these, including an emphasis post on Bradford as well as UV-based methods. If you want to actually look and see how pure is your protein, how many different proteins are in the mixture, things like this, you often run an SDS-PAGE gel, which is going to separate the proteins by size. And then you can stain the gel with something like Comasi in order to see bands show up at the positions of proteins corresponding to how big those proteins were. And so what you want to see with the purification is that as you move on in your purification, you have fewer and fewer bands on your gel, there's fewer and fewer proteins in the mixture, and then ultimately what you want to see is that one nice pure band. Although there can still be multiple bands hidden in that one band, but hopefully it's just your protein. In order to keep that protein that you've worked so hard to purify safe, you're typically going to flash freeze it. So you want to freeze it really, really fast so that ice crystals can't form. Those ice crystals can form when water freezes slowly because the water molecules like to hang out with other water molecules and like link up into this network. And if you give them time, they're going to form this optimal interaction network where they're going to be making these contacts with other water molecules that's going to actually cause them to expand. And so when water freezes, it maximizes its interactions with other water molecules, which requires it to be in this kind of orientation that makes it bigger. And so what would happen is that if you have this water molecules in your protein, so your protein, although it looks like a kind of blobby thing, there's actually water flowing all throughout it. And if that water is going to form ice, so if it crystallizes, if it gets into that network that's going to expand, well, now you're going to like break your protein and things. And you can also have it that the water is going to kind of exclude salts and things like this. So you can have random parts of the solution have higher salt than others. You can have all sorts of different problems if you let it just freeze slowly. But if you freeze it really quickly before the water has a time to link up in those networks, now your protein is going to be safer and your solution is going to be more consistent. And so what we do is we typically flash freeze them in liquid nitrogen, sometimes with a cryoprotectant like glycerol. When you want to actually go and use the protein, there's various things that you can do with it. So if you want to check out the lab techniques page for some techniques you can use, as well as the structural biology page if you wanted to get a look at how you can get a look at proteins. And so hope this was helpful, and happy purifying.